Hello, students. Welcome to the second half of this semester. As mentioned earlier in our announcements, we will be conducting independent student research experiments. So these are going to be group projects that with your lab members that you were previously assigned, you are going to collaborate to design an experiment, and if it is safe and simple enough, you may actually be able to conduct it yourselves. There are a bunch of disclaimers, and I'll go over those additional documents as well. So first things first is that you should verify that you have the contact information of your group members. So for previous labs, you requested group data. It's important that you verify and go back to make sure you actually properly coordinated with your group members. I would probably suggest that once you all have each other's emails, you may request each other's phone numbers, for instance, and put yourselves on a group text thread. There are other apps that can help you as well. And so if you need additional information about that, please reach out to me to my email address. So what I will do now, I'm going to go ahead and share my screen. And then we're going to be looking at the course shell specifically. All right, now you should be looking at the course shell. So as always, before our links to our materials, specifically your lab materials, are under the 1407 laboratory materials link. So as we navigate that, we're going to look at the folder specifically for the independent project. And so I will go ahead and make sure that is available. And once we jump into this, you will see a bunch of information. So first things first, you see attached files. That's going to be information on designing your independent experiment. And then you will have project ideas and links to research guides. So we'll go over those in a little bit. But one of the most important things about this is to understand that this is a collaborative process. Your group needs to get in contact with me to make sure that they understand what kind of experiment that they're doing, that it falls within the realm of Bio 1407, and also that it is scientifically sound, so that you're conducting an experiment that makes sense within the scope of general science. So in general, your groups are about 4 members each. One thing to include specifically is to make sure that each group member, and this is part of an actual assignment, that you are submitting 2 references relevant to your experiment. So the idea is a complete group would then have 8 references. So it's important to collaborate with your group and early on get an idea of what specifically you want to do. And once you get an idea of that, there's actually going to be a form you need to fill out, part of your experimental proposal. So once you discuss it with me and you get it nailed down, exactly what you're going to do, and you find your references, there's some background information that we're going to ask for. And so this talks about these supporting documents we have. And in week 9, you'll actually be doing a presentation over this material as a group. And then here in this second link, we have your resources, additional resources for different organisms you may consider wanting to work with, or different ideas. Alrighty, so if we jump into this first document, go ahead and download that. So we're going to go ahead and share the screen again, but this time the entire screen. And it's going to look a little bit weird for a second, but should clear up once I open up this file type. So just give it a minute, and here we go. All right, so this is actually the first document is a file on designing your experimental research paper, your research experiment. So it asks you a list of questions. First things first is wants you to name the organism, and there's the question, and the question results ultimately in why you're doing this experiment and why you're, or what observations have you seen, what kind of problem are you trying to solve, what makes this experiment something worth diving into. So ultimately, you're looking at a question that corresponds to something that is testable, quantifiable, falsifiable as well, something that you can actually look at and examine in a lab, theoretically. Again, if it's a relatively simple experiment and you have access to those resources, then you will actually be able to conduct it, pending approval, of course, from me in your actual homes. And so the second question asks you, why is it biologically significant, relevant, or interesting? This is important because it ties back to that driving question, what kind of scientific mystery or principle you are trying to solve. One thing to note in the supporting documents you have, to make sure that you're including the actual PDF of the documents, that you have access to a full version of them, and that you are properly citing the articles as well. So you'll provide me with the articles as well as citations for them. And we'll continue to work with null and alternative hypotheses. And so just as a refresher, the null hypothesis represents a condition in which there is no difference between, say, the two data sets or the two means or the two groups, and any differences there is statistically insignificant, and due to random chance or sampling error, it's not due to the actual experimental variable. So the variable you change, that is not why there's a difference between the data set. Whereas the alternative hypothesis states that there is a real, meaningful, and statistically significant difference between the two experimental groups. The effect is statistically real, and for the most part, you can expect it to be meaningful. And so, of course, that doesn't get into the difference between like medically significant or clinically significant and statistically significant, but those are all conversations for another time. And here it asks you in question 5 to think about what kind of results you would get from studying your experimental variables or running your theoretical experiment. And then it asks about the variables that are manipulated in the experiment. Another thing that's important is to think about actual repetitions and quantities. So that will make more sense when I jump into another example. An actual like previous example, previous paper. So one thing to make sure of is that you know how big your sample size is. Um, so how many people you are going to conduct the experiment on, or how many organisms, and for duration of time, how long, and how many trials. Will you repeat this experiment any? And then of course you will look at what kind of statistical tests you will run on your data. We've looked at chi-squared, we've also looked at t-tests, and then we've looked at linear regression. So the idea is that you'll have to use one of those three types of tests to best analyze your data. There are other statistical tests, but you aren't really expected to know them or be able to use them quite yet. And then look at potential sources of error in your experiment as well. Ultimately, you'll be doing a presentation on this and a paper. So let's jump back to the folder. And then you have these additional files. So we'll go and look at the second one. And we'll wait for it to load up. I'm going to go ahead and zoom in on this document. So this basically breaks down some of that same information, but in a more compact way, and using an actual, like giving you additional information. So for here on the specific question, it asks you, like, for instance, what is the effect of fertilizer concentration on lipid production? The big question in this one, it gives you another example, like talks about algae, for instance. And here, variables, you can see how long, quantity, so like actual volume they're using, length of times, days, all that is information you need to include. So specific information, enough that someone could actually reproduce your experiment. So you basically are designing an experimental proposal with enough information that we actually know what you'll be doing in the lab. So if you look back at your previous experiments, these kinds of data points were provided for you via the experimental protocols. So even though you didn't physically conduct experiments, the instructions for the experiments were still provided for you. This time, since you're designing the experiment, you'll provide us and your instructor with the experimental instructions, essentially. All right, we're going to go ahead and I'll let you guys look at project ideas on your own. The library has a research guide, and we'll go ahead and look at that. So I'm going to go ahead and copy that link, once it lets me, of course. Okay, we'll try that again. So as we're looking at the library guide resources, this again is for the Northeast campus. It'll help you link to research journal articles and journal article databases. So here, if you look at this first column, and it'll give you additional information on how to access this information on campus versus off campus. So let's just explore the additional links here. So here you have different journal article databases that will contain different kinds of scientific journal articles. So they're organized a little bit differently, each of their websites. You may find over time that you have a preferred one that you use. Like if you go to all databases, you can look at them by subject, you can look at them A through Z. When I write papers, let me see, he'll link to my favorite one. Definitely doesn't start with a D. There we go. You'll find that this is again for all different subjects. So if you know your specific favorite one that you've used before, you may actually choose to look at it alphabetically and go to the one you're most familiar with. So as you start to use these, you'll find out what kinds of papers they tend to collect and your likelihood of finding something of interest or relevant to your actual experiments. So that's just a personal preference on which kinds of databases you like the best. And if we look at it by subject, probably won't be using the one for art in this experiment. So I'm just going to scroll down. There's one on health and nursing. Let's see. Behavioral sciences. Here we go. And then general science. So here you can, this one has the word biology in its name. That may be a good indicator that you may find more relevant articles in there. Science actually covers a broad variety of topics. So looking at something that says biology in itself could help you narrow down finding papers that you are most interested in for 1407. Like this one says chemistry. You're probably not going to find anything relevant to 1407 in a chemistry link. So just be careful when you click on something that says like science and technology. Know that it applies to all sciences, and it's not just going to apply to biology or Bio 1 or Bio 2. Here is going to be the presentation template. We'll go ahead and look at that once it downloads. Go ahead and open that up. I'm going to try to open it up again. As we're waiting for that to open up, I will go ahead and tell you that I am going to show you some example papers from a previous semester, as well as example, sort of like the condensed down version of the proposal that I'm going to send to the lab staff. And then we'll also help you get some ideas on what students had worked with in the past. Do you love how fast my computer is? I just want to point that out. So in the papers I'm going to show you, I did redact the names. They don't necessarily represent students that did everything 100% perfectly correct, but they are useful for instruction and giving examples. So it looks like it'll be a few more moments, and then we'll get to see this template. If all goes. This is also one of the benefits of posting content asynchronously, is that you can skip forward and fast forward when you watch this later. For me, it's happening in real time, and I don't get the option to fast forward. We may actually need to move on while that's opening, assuming it didn't crash my whole computer. All right, so I'm going to jump into, let me move that out of the way. This is an example of a spreadsheet. So at the end of the semester, we had about, I don't know, 3 groups, so about 4 people in each. So we had about 12 students specifically in my lab section left. We started with 16, so we lost a few students. And so we had 3 different groups. And so the third group, I'm going to look at their paper in a bit, but they actually did an experiment in their house as a group. And so last semester we had the pandemic, so halfway through the semester we went virtual. And so these were actually able to conduct their experiment in their house, so the lab staff did not have to generate data for this group. But these first 2 groups, this is the information I provided. I took it from their lab proposal and placed it in this Excel spreadsheet. And then here, complete means that the data requests were fulfilled and sent out to the students. But this semester, once they are fulfilled, you actually should be getting the data from me. I'll just be passing it along to your groups. And so if there's a question about something that didn't make sense, they were typing back in this document to me, letting me know, hey, something your student proposed doesn't quite work. Will this work instead? So in this group, they looked at the effect of coffee in soil with the idea that caffeine would have an effect on plants. And what's important to note here is how many total plants they wanted, and then the concentration of caffeine in the soil for each plant. And so this group requested really high concentrations, and so I actually requested that the lab staff generate data in more reasonable quantities. And so in reality, this time around, I'll probably just have not passed that information on. I'll stop you in the planning phase and make sure that your data is makes sense from a biological standpoint. They were working last semester. We actually, for the plant labs, worked with Wisconsin fast-growing plants. And as some students learn, fast to sprout and fast to die if you don't take care of them properly. And so that was a big hassle with students accidentally killing off their experiment pretty closely to the start of it. And so that's why large quantities of a substance you're adding to the soil probably shouldn't happen, because you run the risk of killing off your plants before they can grow and mature to full maturity. This other group was interested in seeing how fast plants were affected by different colored light. And so what I thought was interesting about this group is that they looked at non-visible light as well. And so as you know from Bio 1 and Bio 2, the photosynthetic pigments used in chloroplasts will absorb some spectrums of light and reflect others. What's reflected won't be used. What's absorbed will potentially be used. And so the photosynthetic pigments have certain wavelengths of light that will allow peak use for photosynthesis. And so white light, of course, has all of visible light. And so they looked at visible light as well as invisible light from a human perspective. And so there were some fascinating results on that, but that would be spoilers. Don't want to give that away. All right, so here we actually have a presentation template. It finally did open. And so for the most part, this is going to talk about the different categories of things that you included on your proposal, because all that was relevant information. So it's relevant to both designing your experiment as well as conveying it to the general public. So here, again, it's just a title page. So we're going to go through, and again, you don't have to use this PowerPoint, but all this information still needs to be included. The only thing that I would caution you in is making sure your content is readable, reasonable, of course, always, but readable, so you don't do something like red text on a black background. There's probably not enough contrast on that. Really, white text on a black background or black text on a white background is pretty safe. This is obviously really uncluttered and pretty readable as well. But when you're designing your experiment and you're talking about it, you're going to have observations that you've seen just out in nature or during the semester, and that's going to lead you to a specific scientific question that you want to explore. So one of the things that you'll do with your hypothesis is you'll rephrase those observations and those questions into something that can be testable or falsifiable. So a hypothesis has to be testable. There has to be some experiment that can test this specific statement, and there has to be a result that would disprove your hypothesis or fail to support your hypothesis. And so if you can't test it and there's no result that can disprove it, then that's not really a hypothesis per se. That falls outside of the realm of science. So it doesn't mean that the question isn't still important or isn't something that should be discussed. You just may not be able to discuss it scientifically. But since this is a science class, you do need to work with scientific hypotheses or scientific questions. And then, of course, what you actually think will happen. And so here you can talk about your null and alternative hypothesis. Your alternative hypothesis for the case of this experiment will be correlated to what you predict will happen. Here you can include background information about your species of interest. And then you can include different interactions, like how the treatment affects the heart rate or something like that. And then in your experimental design, you'll look at actual concentrations and what is considered a biologically significant concentration. Well, by looking into the literature and your sources, you can actually help figure out what kind of starting concentration and what kind of ending concentration you want. And then depending upon your experimental design, you may want a positive or a negative control. And then in all experiments, you really only want to test one variable at a time so that you can see if that correlates. If you change one variable and there's a change physiologically in your organism or in the variable you're measuring, then there's a chance it's due to the variable you changed. But if you change 3 variables or 2 variables at a time, you don't know which one contributes to your physiological change that you measured. So some students get really ambitious and go a little bit crazy in the amount of variables they want to change or the amount of things they want to test all at one time. But I caution you, it will make your experiment a little bit convoluted and hard to interpret. So if you remember back to the Daphnia experiments, you will want to, we actually looked at heart rate and how heart rate was affected both by different temperatures and by chemicals, but those were 2 separate labs. So we didn't try to change temperature and add chemicals all at the same time, because it would have been difficult to tease apart the physiological changes, how heart rate was truly changed. And you can't really examine that statistically because you're changing many things at one time. And then, of course, you'll want to talk about how you plan on measuring your experimental variables and collecting them. And then, of course, you'll want to statistically analyze them. It's always important to include your references. So if you, again, looked at heart rate and different temperatures, and you did something ridiculous, like you attempted to do 0 degrees Kelvin, and we'll be like, well, that's absolute 0. There's absolutely no way you're going to be able to get that temperature in a lab. And you link it to a source material, and the source material actually tells you how to get to absolute 0. Well, that's kind of useful. Then all of a sudden, if it's doable based off the source material, it kind of makes sense. Of course, that's a random example. Chances are you will probably not be working with absolute 0 in the lab, and you definitely won't be doing it in your home. But all right, so I'm going to now show you some example papers. So I redacted the names from this paper. They were here. This group looked at, again, the effects of light on plant growth. In their abstract, what I specifically like about this is that they included some information about what actually happened to their plants. So we get some numerical data here. Here it's talking about p-values. I would have liked to maybe see, actually it's up here. So they did include how many plants they use. And they talked about average mean, but I think it would have been more useful to actually list the average mean instead of saying you took the average mean. Because sometimes with papers is that when you're at a journal database, you don't always have access to every paper. Your educational institution provides you access and subscriptions to a lot of different papers, but sometimes you have to pay to unlock a full article. And obviously, we're not expecting you to pay. We're just hoping you find a different source at that point that we do subscribe to. But what will happen is that they'll only provide you with the abstract, but they won't provide you with any of the other sections of the paper. But when you have the abstract, you actually can learn a whole lot about the experiment, since it includes their hypothesis, their question to be answered, numerical results, and a pretty solid conclusion on what happened. So for this research paper, we want you to only use papers that you have access to the full amount. but in other classes, potentially, the papers can be really difficult to read and understand if you're not a subject matter expert. But the abstract generally is so short and so concise that emerging scientists and non-experts can get a pretty good grasp on what is happening in the paper by just reading the abstract. Whereas if they read the whole discussion section, they may be relatively confused. The content may be over their head or out of their area of expertise. And so really in your past papers, we aren't strongly requiring you to include numerical data in your abstract, but I personally think it's helpful. And I've encouraged you in my feedback of grading the papers to state what you mean scientifically and provide evidence to back it up. The best way to do that is to actually, instead of saying you calculated the mean, actually tell us what the mean was. Of course, like in the appropriate section, the results or the conclusion section. So here, first thing is you see that they're talking about background information for their plants. And it's really good that they're including sources already, because if it's not common knowledge, you are expected to provide sources to it. And in the methods section, I see things like numerical amount of days, degrees, hours, all of that is important to include. And that's something that you'll have to really decide once you're doing the proposal, because you really don't get the option to stop an experiment halfway and say, hey, maybe I should have designed this better. It's best to think about all those variables in the beginning, because in reality, you would probably finish the experiment and then once you realize where you made the mistakes, design a whole separate experiment that was done a little bit more thoroughly. Here in the results section, again, I'm seeing a lot of data physically in the paragraph, which is important. It's also important to reference your tables here in paragraph form. And so here they do include a table which sums up what they said fairly nicely, and that's good. We need the full explanation as well as a summary. So they didn't rely on only the table caption to convey what's in here. They actually describe it in the paragraph, which is important as well. And then we see the data represented on a figure as well. And then they provide a conclusion, and then they have references here. So we're going to look at an additional paper. Here, this group is the one that actually was able to conduct the experiment in their home. Again, their names were listed here, but I've redacted them. And here in their abstract, you'll pretty much sum up what they wanted to do. So I don't have to wonder what their experiment was about. They actually tell me and they convey it concisely. So they specifically say the purpose of their experiment was to answer the question of whether eco-friendly or environmentally or ecologically friendly detergents are a better alternative than conventional detergents when it comes to how they affect the environment when polluted with wastewater that contains them. So whenever you wash dishes or you wash your clothes, that leftover water generally goes down the drain to the sink, or it gets exhausted out of your house, and that will then go to a water treatment plant, and then that will be tried to be made safe to drink again. But basically, we try to purify water, break down the contaminants. But depending upon where you live, what country, what kind of water system you have available, what kind of infrastructure, sometimes, and also the idea that a more efficient use of some of that kind of water, specifically from clothes and plants, could be recycled in home by itself to say like water your lawn or water your plants. So the presence of detergents, would that negatively affect the environment or positively, even on like a hyper-specific local scale of your front lawn? Would it be damaged if you watered your plants with wastewater from washing your clothes? And so they looked and used marigolds specifically. And again, they're able to conduct this experiment in their own home. They looked at both germination as well as mature plants, and they give their explanations why. They have a list of materials here. You really don't need that in a scientific paper. Like for instance, it's really obvious that we don't really need to know the brand or what kind of marker you use to label your plants, unless it's like some really specific technique that no one's aware of. But in lab, we use markers all the time. So you really don't want to include a raw list. We actually count off for that. And here they have pictures of the plants before they germinated as well as their seeds. And then what they looked like as the seeds were starting to sprout. And because they're looking at things like number of leaves and plant height, we can see how they measure them as well. And then they talk about how they came up with their concentration. And then you look at the end result. The control, which was just water, had the best plants. The eco-friendly detergent had a medium, wasn't as healthy as the control, but it was more healthy than the conventional detergent. And so that's something they talked about. They have data to back it up. So overall, this was a pretty solid experiment. They present their data in a really good way. I wouldn't necessarily be using colorful charts, but again, no paper is perfect. And then again, something like this, there's a lot of information on this chart. I probably would have broken it down a little bit differently, maybe into two separate charts. And the actual data I presented may be in a more condensed format. Just because someone looking at this immediately, it's going to look a little bit information dense. All right, and they included lots of pictures too. These are pictures they actually took, which was pretty cool. All right, and then this was a paper that looked at the effect of coffee grounds in soil. Here they have a pretty concise abstract. They don't really talk about in the abstract any of them what they actually measured. I'm assuming they measured something like plant height, but they don't actually tell me in the abstract. So again, like make sure you're conveying what you actually did thoroughly in your abstract, that the person reading has a pretty good summary of your entire experiment. And so here they go into background information, their methods on how they're going to design their experiment. Oh, this group, let me see. They did pretty high concentrations of coffee, 1-to-1, 2-to-1, and 3-to-1. And so they actually wanted to really look at caffeine, but they didn't have access to caffeine extract, so they actually just used coffee. And then, of course, it's important to make sure they're using one specific brand with a known concentration of caffeine in it. So if they had used a bunch of different brands, then that would have been too many variables to measure at one time. And here it looks like they said, okay, it looks like they measured stem length sizes, so height and number of leaves. And so for their experiment, they really wanted to look at caffeine, but they ended up using coffee instead because it was just practically, it was simpler. And so they had to compare all the different groups to each other. The control to 3-to-1, the 3-to-1 to the 2-to-2, the control to the 2-to-1, and etc. Number of leaves versus coffee concentration in soil. This is a relatively straightforward graph, which I like. They're probably not accurately, well, in this legend here, they tell you what the 1, 2, 3, and 4 is. But here, just glancing at this data, you notice that the control has around 6 leaves, but the 3-to-1 ratio, so the one with the least caffeine in it, I believe, had even higher number of leaves within control. So it's almost as if the actual concentration mattered of caffeine, but the relationship between it wasn't quite what you would think. So a little bit of caffeine, it may be harmful. Well, a lot of bit of caffeine would be harmful. Lesser quantities may actually be beneficial. Not some may be neutral and others may be beneficial. Based off of this, experiment, they didn't conduct the actual experiment. We just used theoretical data for this experiment. So we'd have to then again look at the sources of the theoretical data. So we would have an idea on exactly what happened. And so those original researchers may have seen, who knows what kind of relationship they originally saw. It would be interesting to be able to link it back to the source. Or you would conduct this experiment another time with more concentrations or even different concentrations and seeing if there was maybe even doing linear regression on it and seeing how strongly the data was giving you a linear predictable relationship. And so here again, this last link will give you a little bit of information on how to properly write a scientific research paper, but you guys should actually have a pretty good idea on how to do it since you have or are working on 2 research papers before you actually write this one. So again, the most important thing is to get with me and get with your group and come up with a plan on what you want to test. You can also look down here and look through these links and see if anything piques your interest or think about what you learned in lecture and see if you want to kind of do an experiment based off of that. So that's all the information I have for you guys today on the independent experiments. If you have any questions at all, feel free to reach out to me directly at my email address. I expect to be getting a lot of back and forth communication from you guys during this whole process. So at the end of the day, I don't need to be included in any of your text threads, but once you do need me to look at something, make sure you are including all your group members on the email thread so that I'm not conveying the information to just one person. So, and again, if you have issues with lab members not really responding or not contributing, feel free to reach out to me and we can resolve that on an individual case-by-case basis. So that's all the information I have for you now. Again, I'll see you next time we need a class update.